being able to self-pollinate presents a big advantage for plant fitness because self-pollination is more efficient in spreading the genes of an individual. Because self-pollinating individuals use their own pollen to fertilize their own ovules, the offspring they produce will inherit two gene copies of the parent, which acts as pollen donor and pollen receiver. An individual that only does cross-pollination can either transmit its genes through the pollen which fertilizes other plants, or through the ovules that are fertilized with pollen from other plants. In both cases, the offspring it produces will have only one copy of the outcrossing parent, and the other one will be from another outcrossing parent. In total, selfing individuals leave two gene copies per offspring, whereas an outcrossing individual leaves only one copy. This is called the automatic advantage of selfing to outcrossing. However, selfing can also have negative consequences for the plant performance, because selfed offspring can suffer from inbreeding depression. Inbreeding depression is a decrease in the performance of selfed or inbred offspring relative to outcrossed or outbred offspring. This can be a decrease in the size of seeds or slower growth rate, fewer flowers, or anything else that reduces the fitness of the offspring. You can view these two forces as an opposition between quality and quantity. Automatic advantage increases the quantity of the transmitted genes, whereas inbreeding depression decreases the quality of the offspring. Automatic advantage is a clear mathematical advantage. Transmitting two copies is better than one for the spread of individual genes. It does not matter whether the genes are dominant, recessive, advantageous, or deleterious. But inbreeding depression is governed by more sophisticated mechanisms. To measure inbreeding depression, we need to compare the performance of offspring from the same parent that was produced by selfing and outcrossing. If the performance of the inbred individual is lower than the performance of the outbred individual, we have support for the existence of inbreeding depression. The leading theory about what causes inbreeding depression is the expression of recessive deleterious alleles. Let's imagine that the parent that produces these seeds is heterozygote for a fitness-related gene, with the purple allele being dominant and the gray allele being a recessive deleterious allele. If the individual self-pollinates, there is a chance that it will produce homozygote recessive offspring. In this offspring, the deleterious effects of the recessive alleles will be expressed because they are no longer masked by the dominant non-deleterious allele. However, if the heterozygote individual reproduces with another non-related individual, the chances are that the two non-related individuals have the same deleterious allele are infinitely small. The offspring will most likely inherit at least one dominant functional allele and will thus have higher fitness than the inbred offspring. Regardless of the genotype of the individual, if we observe a decrease in the fitness of the inbred relative to the outbred individuals, we can say that the species, or the studied family, suffers from inbreeding depression. We denote inbreeding depression with the Greek letter delta, and we can calculate it as the difference between fitness of outbred and inbred individuals. In order to make this value comparable among traits and populations with different measurement units and scales, we divide this difference by the fitness of outbred individuals. Because of this scaling, inbreeding depression is a value that is positive and comprised between 0 and 1. The closer is delta to 1, the higher is the inbreeding depression. If delta is close to 0, then there is no difference between the fitness of inbred and outbred offspring, and there is no detectable inbreeding depression. What do you think? How strong does inbreeding depression need to be in order to cancel the effects of the automatic advantage of selfing? Pause and think about this. If inbreeding depression is 0.5 or higher, the performance of inbred offspring is reduced by half. So the automatic advantage of selfing no longer holds. Although selfing transmits twice as many genes, because of inbreeding depression, these genes contribute only half to their parental offspring. Here is an example of Minuartius maecali, an endemic serpentinophyte species that is only found in 2 locations in the Czech Republic. I ran an experiment with this species from 2017 to 2018 in Prague. I had an army of students which spontaneously called themselves chmelaci, translating as bumblebees in English, making controlled crosses in an experimental population of Minuartius maecali. They took the pollen from one anther of one flower and deposited the pollen on a stigma of either the same flower for self-pollination or a flower from a different plant for cross-pollination. The students did this about 2,000 times, which is a very painstaking task given that the flower of Minuartius maecali are about the size of a fingernail and their pollen is white and their pistils are white too. But they bravely managed to perform all 2,000 crosses, and the seeds that were produced were grown in a controlled environment and were measured for several traits correlated with fitness: flower set, number of stems, and plant size. For all 3 traits, you see that their value is higher in cross-pollinated than in self-pollinated individuals. And if we calculate inbreeding depression, we get results ranging between 0.41 to 0.44. This value is below 0.5, which means that if there are no other evolutionary forces in play, it might be more advantageous for Minuartius maecali to reproduce through self-pollination than through cross-pollination.